Hello, it's great to be here presenting. Thank you, Audrey. And also thank you to the AV team and Kai for making this transition, I think, as seamless as it could be given this, what we call winter snowpocalypse, but it really is not too bad, I would say. So I think my talk should be a pretty good complement to the talk that we just had from Dr. Sai. Rather than looking at genetic etiologies of cerebellar dysfunction, what I'd like to do is kind of go over the various reasons why the cerebellum is particularly vulnerable during prematurity and how that may impact neurodevelopmental outcomes. I'll start with kind of a broader overview, and then at the end of the talk, show a model that I've developed in the lab of Dr. Roy Silito, who's my current research mentor, to try to investigate mechanisms and kind of look further into this issue on the side of basic science. So just briefly, kind of the objectives, which will be the structure of my talk, we'll look at kind of the reasons why the cerebellum in particular is anatomically vulnerable during prematurity, followed by the risk factors that have been shown to contribute to cerebellar dysfunction in early prematurity. And then finally, look at the model that we're using to evaluate the outcomes based on the timing of cerebellar injury. So here at the bottom left is the cerebellum. From very early anatomical studies, it was noted that it just feels and looks different, like literally feels different than the rest of the cortex. And, despite occupying only 10% of the brain volume, one of the things that contributes to that different texture, the hardness of the cerebellum, is that it actually houses around 80% of the neurons in the fully developed nervous system. So despite being only 10% of the volume, it actually contains computational power that exceeds the remainder of the cortex and the subcortical structures there. So the cerebellum has a very neat kind of input-output structure. It receives ascending inputs from the inferior cerebellar peduncle, from the spinous cerebellar tracts, as well as the inferior olive, as well as what I would term descending inputs, which come from the neocortex. And these go through the middle cerebellar peduncle via the pontine nuclei. Now, they both project up to the cerebellar cortex, where the computational functions are performed. Then output on the Purkinje cells, as Dr. Sai mentioned, which go to the cerebellar nuclei. You can see on the right here. And those project to the thalamus and other cortical areas via second and third order projections via the superior cerebellar peduncles, which are the main output tract that has been kind of implicated in cerebellar mutism or posterior fossa syndrome. All right, let me get this. Here we go. So now, if you look on the right here, this is kind of a very, pulled back schematic of the cerebellar cortical circuitry. So you have the inputs from the inferior olive and the pontine nuclei projecting to the cerebellar cortex. In blue, you have the granule cells, which are actually comprise the vast majority of neurons in the nervous system. And those converge on, with many other cell types that I didn't show here, onto Purkinje cells, which then output to the cerebellar nuclei. Now, in this review in 2019 by Diedrichson, the point is made that this actually corresponds to the largest information expansion and the largest bandwidth in the entire nervous system. So you have a massive amount of computational power projecting to the cerebellar cortex and then back out to the rest of the cerebellum. Now, what this means in practice, and this is something that Dr. Sai alluded to quite a bit, is that there is actually functional connectivity, which refers to correlated activity between the cerebellum and essentially all areas of the neocortex. So you can see both in the primary sensory and primary motor regions, you have quite a bit of activity, but it's not restricted to those areas. In fact, even in limbic regions and the prefrontal cortex, here you can see a nice demonstration of that right crus with 16 in yellow there and the prefrontal cortices. But you do have this massive functional and anatomic connectivity. So how does this relate to prematurity? Now, you can see here at the beginning of, when we first start seeing premature babies being viable at 24 weeks, the cerebellum is this very small structure. Over the course of the third trimester, you have a massive expansion of the cerebellar volume as well as cerebellar surface area, which is achieved by the birth, differentiation, migration, and wiring of the cerebellar cortex, including both granule cells as well as Purkinje cells. So this period, the third trimester, is one in which there's this massive expansion of cell density and wiring, and this gives this kind of a particular vulnerability during the rough transition from intrauterine to extrauterine life for premature babies. So just briefly recapping, we have seen that the cerebellum has this massive anatomic connectivity to the cortex, which is reflected in this functional connectivity that we can see through fMRI studies. And finally, most importantly, in the context of this talk, the cerebellar development actually largely begins at the beginning of the third trimester. So how does this relate to the development of the cerebellum when we have extreme prematurity? There have been quite a few studies, mostly in the past 15 years, that have started to understand this phenomenon. Now, briefly, it's not surprising or unusual to hear how much of an impact prematurity has on neurodevelopmental disability. So up to 50% of children born early preterm, which is usually between 24 and 32 weeks, will experience significant deficits in all behavioral domains. So this includes cognition, socialization, behavior, attention, and amounts to over around 20,000 children per year who experience neurodevelopmental disability during prematurity. And importantly, this number is only increasing as our neonatologists are getting better and better at helping children through this rough transition that occurs when they're so young and vulnerable. Now, we know, pretty much all of us, that the associated finding with early prematurity is IVH and the resultant periventricular leukomalacia. That, in fact, is the most common finding in this population, the early preterm population. However, in addition to that, cerebellar insult has been found to be a very significant contributor to the neurodisability associated with prematurity. Why is this so? So one of the key mechanisms is cerebellar hemorrhage. So independent of any findings in the supratentorial space, if you have isolated cerebellar hemorrhage, there's a massive predisposition to severe impairment, and even more so, minor impairments. But these two meta-analyses that were recently published by Hortensis and Villamor-Martinez have kind of outlined this nicely. So even when you have small punctate bleeds, you can see impairment in up to 20% of the children who were born premature. Now, when you have larger bleeds or you have involvement of the midline, the massive midline structure, the cerebellar vermis, those numbers go up to almost, in some cases, up to 90% when you're talking about vermal involvement. So the reasons for this aren't really well understood, but there's a lot of different speculation out there, most of which relates to the hemodynamic instability that occurs during prematurity and potential watershed areas that lead to infarcts in the cerebellum, then leading secondarily to hemorrhage. So a second reason, and one of the ones that I kind of found the most striking as I started to get into this literature, is the association of the prematurity with hemosiderin deposition in the posterior fossa. Now, as mentioned before, the most common finding in prematurity and children with disability associated with prematurity is periventricular leukomalacia, which is usually a reflection of germinal matrix hemorrhage or IVH. We grade that 1 through 4, where we see very mild changes all the way up to cystic changes in the cortex and the periventricular white matter. Now, independent of the degree of hemorrhage that the child experienced, you can see associated cerebellar hypoplasia. And mechanistically, what's thought to be going on there is that just solely based on the anatomic position of the cerebellum, this kind of dependent position in terms of CSF flow, that you get blood overlaying this massively expanding anatomical structure, and then the physical irritation and siderosis that occurs leads to alterations in the development of the cerebellar cortex. Here you can see in these images, not only hypoplastic cerebellum, you can also see evidence of the hemosiderin deposition. And this is something that we can see on multiple different sequences. And it's probably the key reason why we have an association between prematurity, IVH, and cerebellar hypoplasia. However, in addition to simply the association between IVH and cerebellar hypoplasia, there are other reasons that we have hypoplastic cerebellum in early prematurity. So here's a study published in 2005 that kind of shows, several different things. So in the white boxes, you can see just generally in kids who don't have any kind of cerebellar injury, the degree of expansion that you see in the third trimester, going from 27 weeks to about term gestation. In the white boxes, you see this kind of, very large increase. Now, when you have abnormal MRIs, and that would include supratentorial injury, you see a much shallower increase in cerebellar volume. Not shown here, but described in the study, is that even in the cases where you have a normal MRI during prematurity, you actually see about a 3 cc reduction in cerebellar volume, and that was found to be significant here. The mechanisms for this include, the different factors associated with kind of an extrauterine third trimester. So there are different ideas out there, as I alluded to with the cerebellar hemorrhage, hypoxia, ischemia, and other associated issues with hemodynamic instability in early prematurity contributes probably greatly to this phenomenon, as well as glucocorticoid exposure, undernutrition, or changes in the nutritional status that are, go with the transition to extrauterine life. And then, kind of a very interesting idea that's not very mechanistically well understood, which is the loss of trophic support from changes in connectivity. This is referred to in the literature as cross-cerebellar diasthesis, and leads to altered network connectivity. Potentially mechanistically, some things that we see with those functional MRI studies that were alluded to in the previous discussion. So here briefly, just stepping back, we first saw the degree of expansion and cell birth and wiring that occurs in the first trimester, and then now the predisposition of the cerebellum itself in the third trimester to suffering insult in the context of this massive expansion and increase in surface area. So finally, I'd like to talk a little bit about the work that we've been doing in the Silito lab to understand, how and the mechanisms by which altered cerebellar neurotransmission may lead to changes in neurodevelopmental outcomes. So briefly, I'd like you to recall the cerebellar connectivity that we discussed. So you have these inflow tracts, afferent projections from the pontine nuclei in the inferior olive going up to the cerebellar cortex. They project their synapse either directly onto Purkinje cells or other neurons, and including the granule cells, which are the most numerous neuron in the nervous system, do the computational processing that the cerebellum conducts, synapse onto Purkinje cells, which send inhibitory projections, GABAergic neurotransmission, onto the cerebellar nuclei, which then project to the subcortical structures and via those structures to the neocortex. So what we wanted to do is develop a model where we could understand, okay, if we silence cerebellar function early in development versus later in development, what kind of impacts do we see on the behavioral outcomes in mice? So what we did is we used a TET cassette where we can have tetracycline in the diet, preventing Cre-base recombination. When we have tetracycline in the diet, we have normal GABAergic neurotransmission from the Purkinje cell. When we remove tetracycline from the diet, what we do is we excise the vesicular GABA transporter, specifically in the Purkinje cell population, and then mechanistically and functionally we can silence cerebellar output. So using this model, what we wanted to do was interrogate the effect of the timing of cerebellar injury, or in this case, induced cerebellar dysfunction, on the behavioral outcomes of the mice. So what we wanted to do is pick 2 time points initially and see what the effects were. So first, we're able to silence in gestation, and so in these mice, the mating pair is never given tetracycline. So from the very initiation of Purkinje cell differentiation and Purkinje cell wiring, their neurotransmission is silent. We then wanted to compare this to a later time point. So the term development in mice is approximately equivalent in mice to postnatal day 21. So if we take tetracycline out of the diet around the time of weaning, this would consist our late time point where we silence the cerebellum after the majority of cerebellar expansion has occurred. So late silencing, what we found, if we turn off cerebellar neurotransmission around the time of weaning, has a fairly subtle effect on behavior. And here we'll show a video. So on the right here, you can see the animal, the experimental animal that has silenced neurotransmission. There's, a little bit of imbalance. And on the left here, you can see the wild type animal, just for comparison. So we're continuing to characterize this behavioral phenotype, but just kind of qualitatively, you can see that there is a subtle but noticeable effect on the behavior. Now, if we look at early silencing, where there's really no Purkinje cell neurotransmission, even from early in behavior, you can see a very more severe behavioral phenotype. If we can see this video. So if you look at the tail, you can kind of see a kink there, which reflects dystonia. Even in the legs, they'll have dystonic movements that prevent a normal gait. There's some ataxia that you can see, and what from here, what we want to do is begin to quantify these behavioral phenotypes and then try to understand, are they restricted to motor behavior, or do we see them in other behavioral domains as well? So to test the gross motor behavior, we use the accelerating rotor rod paradigm, where we place the mice on this rod that's spinning and that accelerates over about a 5-minute period. And then we do this test over 3 days. So what we found is the early silencing, as you'd expect, given their severe behavioral phenotype, they don't perform nearly as well as their littermate controls, kind of reinforcing this gross motor phenotype that we see. We're currently working on many different assays to kind of further quantify the dystonia as well as the ataxia and gait abnormalities in these animals. Now, if we perform the same assay where the silencing of the cerebellum was done much later after the full expansion of the cerebellum, we see that they perform this gross motor task just as well as their littermate controls. Now, the second really thing we want to look at, just broadly speaking, is some non-motor functions. So the first assay that we kind of tackled was looking at the open field assay, which people in the literature in the mouse world have kind of used as a correlate for attention deficit disorder. And if we look here, we see that both in the early silencing cohort as well as the late silencing cohort, even despite their either significant or subtle gross motor phenotype, they're actually much more active than their littermate controls. Showing that there probably is this, in addition to the motor phenotype, which we would normally associate with cerebellar activity, there's certainly non-motor aspects to it. And we're currently in the process of performing other assays to evaluate non-motor behavior. So kind of moving forward with this project, which is just kind of in its beginning, is to expand the characterization of the behavioral deficits that we've seen to look at gross motor, social, as well as potentially learning and memory assays. Of course, having kind of established, I think that this model is going to be a pretty insightful way to understand how cerebellar function contributes more broadly to cortical function. We're going to look both at the electrophysiological signatures in the cerebellar nuclei, and then hopefully move out into the cortex to understand kind of on a mechanistic level what's going on. And then finally, and importantly, maybe to start to get at this idea of the trophic relationship between the cerebellar circuit and other subcortical and cortical structures, just looking more in detail at the anatomical effects, both in the cerebellar cortex as well as the inferior olive, and potentially moving into cortical structures. So, here I think the findings that we have, even just in the early experiments that we've done using the synthetic model for cerebellar disruption, is that early disruption of cerebellar signaling does in fact lead to a very severe behavioral phenotype. But the timing of the disruption, if it's early, we have this really significant gross motor phenotype, whereas late dysfunction, whether it's already that we've established cortical networks or if there's some other mechanism, that late silencing of the cerebellum doesn't have the same, kind of broad impact on gross motor development. And then finally, the timing of the disruption leads to a kind of discrepant phenotype where we have very similar levels of hyperactivity, but then very different gross motor phenotypes. So, I hope that I've covered the objectives that we had. We understand, the reason anatomically the cerebellum is vulnerable, mechanistically kind of what's going on, and then starting to understand how the timing of this disruption leads to kind of discrepant phenotypes, and that can be very severe when you have early disruption of cerebellar function. I'd like to thank the division, TCH, and the NRI, which are great places to work, and of course, the lab that I'm in, Dr. Roy Silito, who's the K08 award mentor, has just given me a great opportunity, really introduced me to the cerebellum, and sparked the project from the very beginning, really sparked my entire interest in cerebellar development. And then some excellent colleagues in the lab, especially Dr. Amanda Brown and Dr. Micah Vanderhayden, who have published really interesting work, kind of more in detail on how cerebellar circuit development leads to behavioral phenotypes. I'd be happy to take any questions.
Dr. Gill, another tour de force. Thank you for that fascinating talk. I think we're all learning so much about the cerebellum this morning.
Right.
So a couple questions from the audience. One is the cerebellar volume loss disproportionate to other brain regions?
Yeah, so in the Limperpula study in 2005, I think the number they had was it was like 177% expansion of the cerebellum, normalized to 100% of the cortex, and then you do see a steeper drop off or flattening of that curve during prematurity, and I think this is because kind of what I tried to highlight in the first section there, that really a lot of the neurogenesis in the cortex, at least the gross neurogenesis, I think is occurring prior to the beginning of the third trimester, even though you still have kind of the sulcation foliation occurring in the neocortex. It's kind of a fraction of what's occurring in the cerebellum during the third trimester.
Another question from the audience. Do you see social deficits in your model, or do you expect to?
I will expect to, I think. We're in the process of doing the three-chamber assay, which is one of the assays for social interactions. My feeling is that this hyperactivity phenotype is probably just the beginning of the so-called non-motor phenotypes that we'll see. But we're in the process of doing those studies right now. I think the interesting question will be are there phenotypes that emerge with late silencing that we don't see with early silencing? Or is it just a kind of gradual decrease in the, so quote unquote penetrance of the phenotypes we see? So we start maintaining certain functions like gross motor functions, but then the deficits in other domains are kind of persistent across depending when you silence it, or do we see kind of a shift in what the cerebellum is doing in terms of modulating cortical behavior, where we uncover new phenotypes with later silencing that are not apparent with the early silencing? So that will be a really interesting question that I hope we can address.
One more question. How do you measure cognitive abilities in a mouse if their motor abilities are so impaired?
Yeah, I think that's a very good question. As you could see with the two assays we did there, that even with gross motor dysfunction, we still have a decent amount of activity. So there are some assays I think that will be hindered by the gross motor phenotype. There are other assays where I think you may be able to see those phenotypes. I think that's something that we'll kind of tackle as we start performing the assays to see if their function is compromised to a degree that we really can't get good results. I think it's an interesting question also with the difficulty on the clinic when you have severe quadriplegic CP in understanding what the non-motor function of some of those children are. When if you have an inability to speak and you're unable to execute motor function, we don't really have a great understanding of how those children are doing cognitively. So I think it's something that we have difficulty with in our mouse models, but I think it's something that's clinically challenging as well.
That's a fantastic point. Well, thank you so much. I'm going to make some announcements now before we move on to our next speaker.